Welcome to ISL Enabled Virtual Lab of CSIR.
Welcome to Food Savvy, empowering students with knowledge on science and security. In this video, we will explore crucial aspects of food safety, including the risk of water contamination, the impact of chemical pollutants, and the danger of bacterial contamination. Join us as we delve into how these factors affect our health and discover essential practices to ensure safe and sustainable food choices. First, we will move to water contamination under quality testing. Water quality can be assessed through several factors including pH, conductivity, total dissolved solids, chloride concentration, and calcium levels. In India, water quality standards are provided and monitored by various authorities such as Central Pollution Control Board, Central Groundwater Board, and the Department of Water Sources, River Development, and Ganga Rejuvenation. First one is the pH of drinking water. The pH of drinking water can be measured using a pen type pH meter. The pH of most drinking water should fall within the range of 6.5 to 8.5. For this experiment, we use 50 ml of tap water and measure the pH using a pH meter. In this case, the meter shows a pH of near 7.4, which is within the acceptable range of drinking water. Next, we will move to conductivity of drinking water. Conductivity measures the water's ability to conduct electricity and provides an indication of the dissolved mineral content. When a conductivity meter is dipped into the tap water sample, it shows a reading of 15, which then multiplied by 10 gives a conductivity of 150 microsiemens per centimeter, which is not within the acceptable range for drinking water. That indicates poor quality of water. Next, we will move to total dissolved solids, that is TDS. TDS refer to the total amount of dissolved inorganic and organic substances such as calcium, magnesium, potassium, and sodium in the water. The acceptable TDS range for drinking water is 50 to 150 ppm. Using a TDS meter, the value of water sample reads 51 ppm, which is then multiplied by 10, giving a final value of 510 ppm, which exceeds the recommended range. Analysis of chlorine ion in tap water. To analyze the chloride concentration in water, a titration method is used. The following materials under reagents are required for the titration experiment: silver nitrate, tap water, potassium chromate, 1 ml pipette, conical flask, a burette filled with silver nitrate solution. 20 ml of distilled water is added to a conical flask, followed by 1 ml of tap water. After shaking for uniform mixing, a few drops of potassium chromate are added, turning the solution into yellow color. The solution is titrated with silver nitrate until the color changes to brown. By using V1N1 is equal to V2 into formula, we have estimated the value of chloride in tap water, that is around 710 ppm. Then we are moving to analysis of calcium ion in tap water. For this titration, we prepared the following materials, that is standard EDTA solution, tap water, Ediochrome black tea indicator, ammonia buffer solution, conical flask, pipette, and burette filled with standard EDTA solution. For the titration, 20 ml of distilled water is taken in conical flask and then 1 ml of tap water is added. Mix well for uniform mixing. Then EBT is added, shaken well, and few drops of ammonia buffer is added. Then the solution color changed transparent to red. Then we are titrating against EDTA until the solution color changes to blue in color. To determine the calcium level, V1N1 equal to V2N2 formula is used and the level of calcium in tap water found to be 400 ppm. That's all about water contamination, and we'll move to chemical contamination. The obtained value indicates that the water is of poor drinking of quality. These are the final results of our experiment. Here we have compared with the standard value other than pH, all the values showing the water is not drinkable. Effective solutions for reducing TDS, conductivity chloride and calcium in water, reverse osmosis, distillation, deionization, ion exchange and electrodialysis. This concludes the analysis of water contamination. Next, we will move on to chemical contamination analysis. Chemical contamination in food can occur at various stages of the food supply chain, including preservatives, coloring agents, flavoring agents, sweetening agents, stabilizing agents that can cause serious health hazards, including cancer, neurological disorders, reproductive issues, etc. Ever questioned if your food is truly natural? Say no to unhealthy food and choose nutritious alternatives. Prioritize natural, unprocessed options for better health and sustainability. So, it is very important to do exercise, drink water, and eat healthy, save food, save people, remove poverty. Now, let's go to the next session. As we know, several microorganisms are contaminating our food, which causes serious health hazards like diarrhea, vomiting, fever, stomach cramps, etc. So, let's detect microbes in contaminated food. As we know, health is wealth, and it is very important to ensure the safety of our food. Here, we use the sequential experimental steps to examine microbes in spoiled food. Now, let's explore the experimental session. Here, we are using nutrient agar to examine the presence of microbes in food. We are examining spoiled food items like vegetables, fruit, sweet, meat, and rice to detect microorganisms that causes food contaminations. Now let's see the collected specimens. This is the spoiled food, vegetables, sweet, rice and meat. To detect microorganisms, small amount of specimen is taken in a cotton swab and streaked over nutrient agar. This process helps to identify the microbes that grow in each spoiled food item. Here, the nutrient agar helps the streaked microbes to grow in a suitable condition so that we can observe the specific microbes that spoiled the food. Further, store the streaked agar plates at 37 degrees Celsius for 24 hours and sterilize the workplace to avoid external contamination. After 24 hours, we can observe the sample stored in the incubator and monitor the respective contaminants. Here, in the bare agar plate, no microbial growth was observed, but the vegetables, sweet, rice, meat, and fruit were highly contaminated with microbes. Here you can see different types of food items are contaminated by different microbes. From this, we can understand that the intensity of food contamination by microbes is too high to cause the several diseases. This contamination can only be prevented by safe and healthy practices like eating well-cooked food and avoiding fast food. Also maintain a good hygiene practice like washing hands and drinking boiled water. Always try to keep the food items closed or ensure proper storage to prevent unexpected spoiling of food by airborne pathogens. Always make sure that the food you eat is not expired to cause any health issues because expired food items causes serious health hazards. If the expiry date of the food is over, the rate of microbial growth will be more on it. So let's monitor the growth microbes in the strict plates after 48 hours. This is the microbial growth after 48 hours of incubation. It is essential to keep our food safe, so let's check methods to prevent food contamination. We are developing several food packages for extending shelf life and food preservation. Reinforced silica, reinforced PLA was developed with active packaging properties like antibacterial, antioxidant property, sensing property, barrier property, and UV blockage for food preservation. Safe seal, safe meal.